Okay, this question is the Stop and Think 5.2, and now we're going to read it and try to answer these two questions. Okay, so here we have a recessive allele for four genes, A, B, C, and D, and are clustered in a small region of a chromosome, but the order is unknown. Okay, so we have recessive alleles, and how many of them? Four, as they show in this schematic, but we don't know the order. Okay, so we know that they're there, but we don't know which one is first, second, third, and fourth. Which one are in the center, which one are in the extremes. So how we solve these problems? To solve that problem, what they generally do in genetics is they use mutations that are made heterozygous with each of two deletions, and they have two examples of deletions. Yeah, one that have some of those alleles and another that have another type of alleles. Okay, and later we will talk about that. And they ask these two questions: which genes are at the ends of the cluster, and which genes are compatible with which gene order are compatible with the deletion data. So how do we know which one are at the end and which one, and how we organize those ones? So based on those deletions that they made. So before we focus on the deletions, that it will be, we will make a small genetic map using the deletions. Let me explain a little bit the beginning of the question. So they talk that there are mutants that are analyzed by being made heterozygous, which each of two deletions. Okay, and why that part is important? Something very exciting about evolution and genetics is that, I don't know if you remember, that every time when we start talking about Mendel, we say, "Oh, this is a homozygous dominant, or a heterozygous, or a homozygous recessive." But the question is, why we always have two alleles of the same gene? It's not like why we need the extra one. And some of people say like we have an extra because they can work as a backup. So if we don't have this gene, this allele, it went bad. We have another one that, and in many cases, this one will be more than enough to make the organism viable. And for example, in cancer, you don't need to have one mutation in some genes. Sometimes you need to have the two alleles, to have mutation in both alleles to actually cancer to start appearing and to produce those overgrowth. So we don't know we have why we need two alleles, but what we know is, and there could be many reasons, but this definitely to have a backup is very useful. And in genetics, this is super useful. Why? Because many times you can make something that is, when it's homozygous, that is lethal. Yeah. So for example, here we have a deletion of this first gene, and that could be lethal. If we make it heterozygous, it will be enough to make it viable. And you can start playing with that in different ways. So for example, you can have a double heterozygous. So you can have a deletion here and a deletion here, and this can start helping you to start tracking the genes, to start saying, oh, maybe is it still viable in this way? What type of phenotypes do we get when do we get a white-eyed when we break this gene? And when we break this one, do we still have a white-eyed, or do we change the white-eyed, or what happened here? So this can give us some way to play, to try to organize, to start mapping the genes. Okay, so now let's try to answer the question of the 5.2 question, now that we know this information. Okay, so now that we know why they make it heterozygous, why it was important, and why we use deletions, now let's focus on the two types of deletion. The one that is in green, that uncover the alleles A, B, and D, and the second one that uncover B and C, B, C, and D. And with that information is enough to know which one are in the extremes and which one are the order of those genes. So let's go back into the genetic map. So here what they are asking you is now to use only mutation, but to use deletions for a mapping, for genetic mapping, or for gene mapping. And here I give you an example. So here what they generally do is to take the wild type, and then you start creating fruit flies that have different type of deletions. And in that way, you can start analyzing phenotypes. So, for example, this one produce one that the flies have a curly wings. This one produce curly wing plus white eye. This one produce both things. So you can start saying, oh, maybe these two genes are in charge of this, while the other ones are in charge of the other ones, and you can start organizing which one are in the extreme or which one are in the center. Yeah, and it's a little bit similar to this application this game on your cell phone, that you try to play, okay, which one is first, which one is second, and you just start tracking patterns. In this case, you get this type of maps. Yeah, for example, here you can see that in all the cases, this seems to be in the extremes. Yeah, independent of when you have all these mutations, all this deletion, all these strains of flies, in all the cases this is still available and it seems to be in the extreme. Yeah, and if you see, you can when you have B and C are generally seems to be always together when you create that. So you can start saying, and you can create the more mutations that you have, and the more finer, the more information that you get. So you can start playing with this in the same way that you play in the game to start getting the best maps. So now let's see the two examples that they gave you. So they give you one that you have A, B, and D. So A, B, and D, and you only have C. So you through deletions, you discover that there is, when you remove this one, there is one marker. When you remove this one, there is a marker. When you remove this one, there is another marker. And here, the second deletion, you get B, C, and D. So you have these two things that are A and C are not in common. Yeah, and this is very important. Well, for example, B and D are in common. Yeah, and in the same way that in your game, that this one I call it A, B, C, and D, it doesn't mean that A is first or B is second and C. Yeah, it doesn't mean that. You have to organize it. Yeah. So let's start playing with those. So the answer here, it will be that B and D will be together while A and C are in the extremes. And the second deletion, it will be A is in one of the extreme, and B and D are here in the middle, and C is in the extremes. Yeah, so this is the part that we missed, while in the other one we missed that part. Yeah, so the line represents the section that we missed, and here the line represents the section that we missed. Yeah, there are different ways to represent that. It could be also with minuses and pluses, depend on the example of your textbook, and they will have a different notation that they will have to explain. But how do we come to this conclusion? If you see here, B and D are in common. Yeah, so the most likely is that they are close together. Yeah, because if you break one, you're breaking the one that is closer to that. Yeah. On the other hand, so B and D are always present. Yeah. When one is broken, the other one is broken, so it means that they can be close to each other. On the other hand, A and C sometimes are present, sometimes are not, so maybe they are not, they are in the extreme. So here, for example, if you see A, these three are here, but A is not there, and here A, B, and D are together, but C is not there. So if you see in the green one, we just broke this part, while in the other one, we broke the other part. So yeah, so based on on these two deletions, the most likely answer will be that A, B, and D that the first one will, in the two extremes, you will find A and C, and B and D will be in the center. Okay? Or it can also be, it can be C and here D and B, and A will be in the center. Here we don't have a specific order. It can switch sides. It can be in the reverse, but based on the information that we get, this is the most likely configuration. So answer. So based on this information, the genes that are at the end of the cluster are A and C, because both deletion uncover B and D, this one. And the two possible gene order could be A, B, D, and C, and A, D, B, and C, because they are compatible with the deletion data. Again, if we have the more mutations, the more strains that we can test, we have more information to confirm that. But so far with that information, this is the best answer, the more parsimonious answer that we can get. And if you have any doubt, just let me know.